Hello friends, welcome to my course on microbial biotechnology. We are in module 2. Today we will be discussing about life cycle of representative groups of microbes. Particularly we will be discussing the life cycle of bacteria, then algae and also the life cycle of fungi. So, let us start with the life cycle of bacteria. So, here you can see a division of the bacterium Acromatium, where it is getting divided into two cells. How does this happen? We will be discussing in this section of this lecture. So, what is the reproduction in bacteria and how it happens? All bacteria reproduce through binary fission, which is an asexual process where one cell splits into two identical daughter cells, as we have shown in the earlier introductory slide. So, if you see here one single cell, the final product will be two daughter cells, and what happens in between we will be dealing with it right now. This asexual method allows for rapid population growth, doubling every cycle of cell division, as each daughter cell inherits the parent's genetic material and can immediately begin dividing. Under favorable conditions, bacteria can quickly multiply, leading to exponential population growth. So, cell division involves first the partitioning of the nucleic acid or the genetic material equally between the two daughter cells, and then there is formation of an septum, after which the two daughter cells are separated out. So, if you look into it in more detail, say this is the starting point, or this is the mother cell as well as the daughter cell, which has become matured. There is a placement of a division site where there will be assembly of a Z ring. So, this is the mid cell division site. So, you see the FTS Z polymerization, which gives to a dynamic Z ring. Then there is assembly of divisome, activation of cell wall assembly, and we can see that there is a cell constriction over here taking place, and then after this there will be initiation of separation. This constriction will keep on growing, so as you can see over here. So, it is growing inside to the cell, and then finally, there will be cell separation in the final steps and hydrolysis of the cell wall and fusion of the membrane, giving rise to not one, two daughter cells. We draw here only one for sake of simplicity. So, this cycle goes on and on. So, for every cycle, there one cell will produce two cells. What are the steps in binary fission? The process of bacterial binary fission involves the following steps. Number 1, as I have already told, DNA replication. The circular bacterial chromosome is replicated, ensuring each daughter cell will receive a copy of the genetic material. Then there will be cell elongation. The cell elongates and the two DNA molecules move to opposite poles of the cell, attaching to the cell membrane. Then there is a septum formation, which is a partitioning, which begins to form at the cell's midpoint, dividing the cell in two. Then there is a completion of the division. The septum will grow inward, fusing the membrane and cell wall, creating two separate daughter cells, each with identical DNA. And finally, there is a cell separation. Enzymes break down the cell wall at the division site, fully separating the two daughter cells. Elongation of the bacterial cell. The elongation of the bacterial cell is a crucial step before septum formation and division, ensuring proper distribution of genetic material. During this phase, new cell wall components, including peptidoglycan, are synthesized to allow the cell wall to expand. Some of the key proteins which are involved in the cell elongation include MREB, which is an actin-like protein that helps determine cell shape and coordinates cell wall synthesis by guiding enzymes involved in peptidoglycan synthesis. Then we have MREC and MRED, which stabilize and organizes the MREB filaments. Then there is a RODJ protein, which ensures proper localization and function of the MREB. And then there are PBP2 and PBP2B, which are enzymes that causes peptidoglycan cross-linking for cell wall integrity. Then there is FTSH, which helps maintain the cell envelope during elongation. These proteins will work together to ensure uniform cell wall expansion and proper elongation before division. The septum formation. So, this is the septum formation pre-stage, where there is FTS jet polymerization, as I have already mentioned. Let us look into it how these happen. So, the bacterial cell division requires the regulated polymerization of FTSZ into a dynamic cytokinetic ring called JAD ring, which recruits a dozen other proteins involved in division. This assembly of proteins forms the divisome, as we have shown here in this picture, which helps coordinate the remodeling of the cell wall and the septal ring constriction, finally leading to the division of the cell into two daughter cells. The key steps involved in this process: number 1, FTSZ polymerization. FTSZ is a tubulin-like protein, forms the JAD ring at the fusor site of division. This ring acts as a scaffold for recruitment of other division proteins. Divisome assembly. Once the JAD ring forms, a series of proteins FTSA, GIP-A, FTSW, FTSI or PBP3 and others are recruited to form the divisome. These proteins play crucial role in cell wall synthesis, regulation and coordination of constriction. Activation of the cell wall synthesis. The divisome activates penicillin binding proteins and other enzymes that synthesize the remodel the peptidoglycan, enabling cell wall constriction during division. Then the cell wall constriction and separation will happen. The combined action of the JAD ring, cell wall synthesis machinery and other regulatory proteins lead to the inward constriction of the cell membrane, eventually pinching the cell into two separate daughter cells. So, if you look into the binary fission of bacteria overall, we can see that replication and segregation of the nucleoid happen, and then there is a polymerization of the MREB proteins, and then these are the MREB subunits, as you can see over here. Synthesis of cell wall and elongation of the cell is happening. So, you can see here this is the size of the cell and here it is getting elongated, and these are the sites of cell wall synthesis. This is the FTS JAD subunits, and this is the MREB filament, as you can see. Then the polymerization of the FTS JAD proteins to form the JAD ring in the center, which we have discussed in the earlier slides. And then this constriction is happening in the JAD ring, and then the completion of the cell division, and finally separation of the daughter cells, which are complete in every aspect. Let us now discuss about the life cycle of algae. So, here you can see the colonial algae, Palmodictyon viral in this picture. So, the life cycle of algae refers to the sequence of events from one generation to the next. Sexual reproduction features an alternation of generations between haploid and diploid stage. So, you have the haploid phase here and the diploid phase here. So, from the haploid it becomes diploid, and from the diploid it becomes haploid. So, this is a general scheme for alternation of generation. There are 5 primary types of life cycle in algae. Number 1 is the haplontic life cycle, then diplontic, then there is a haplodiplontic life cycle, there is a haplobiontic life cycle, there is a haplodiplobiontic life cycle. We will be discussing about these various life cycles one by one. Let us start with the haplontic life cycle. So, before we start, just notice this haploid phase here. which is maybe around 75 to 80% of this charge circle, and there is the diploid phase. And as we have discussed, there is an alternation of generation between the haploid and diploid in algal life cycle. So, this diagram shows the fraction of time the particular algal species is in a particular phase. How much more time it is spending in haploid phase and how much time it is spending in diploid phase can be seen from this colored circle. So, this is the gametophyte. Okay. At the end of the season, gametophytes will generate haploid gametes. These are haploid. So, their number will be n, which fuse to form a diploid zygote. So, there is a fusion over here. So, the zygote is getting formed. So, this is the diploid phase, as you can see over here. So, the zygote undergoes meiosis to produce meiospores that will germinate into finally gametophytes. This cycle is seen in many green algae, charophytes and red algae like Bungia. So, this is the haplontic life cycle. Then we have the diplontic life cycle. Here you can see that compared to the haplontic life cycle, the hap comes from the dominating haploid phase, and here the diplontic comes from the dominating diploid phase. Nevertheless, both the phases are present. So, in the diplontic life cycle, the dominant phase is diploid or the sporophyte, not the gametophyte as in the earlier case. So, sexual reproduction here involves the production of haploid gametes by meiosis in specialized sex organs. These gametes fuse to form a diploid zygote, as in the earlier case of course, which grows into the diploid plant body. Unlike the haplontic cycle, there is no true alternation of generations. The diploid phase dominates and the only haploid stage is the gametes. The life cycle is found in diatoms, certain green algae and some brown algae. The next phase is haplodiplontic life cycle. So, you have both the hap and the dip here. So, in these haplodiplontic life cycle, two distinct generations alternate, showing the true alternation of generation. This cycle includes a diploid sporophyte and a haploid gametophyte. So, this is the sporophyte phase and this is the gametophyte phase. So, there are two types, isomorphic. Both generations are morphologically similar. We cannot separate out the sporophytes from the gametophytes. And then there is the heteromorphic. The generations are morphologically distinct or different. This life cycle is found in groups like ulvales, then cladophorales, as well as Ectocarpus and Dictoeta. So, these sporophyte is diploid and these gametophyte is haploid. Then we have the first subclass of this type of haplodiplontic life cycle, the isomorphic haplodiplontic life cycle. So, you can see here the mature sporophyte and gametophyte, they all look similar. So, the sporophyte and the gametophyte are morphologically similar. And now, one more thing to notice here is the gender of these gametophytes. One is a male and the another is a female. The zygote develops into a sporophytic thallus that produces meiospores in sporangia. These spores germinate into a gametophytic thallus which produces gametes in the sex organs. So, this is the male gamete, and this is the female gamete, and then they fuse, or there is the syngamy. The fusion of these gametes forms a zygote that develops into this zygote will develop into a diploid thallus, and this thallus will give the mature sporophyte. So, we have these type of life cycle in ulvales, cladophorales, ectocarpels, and dictyotales, and also as well as some red algae. Next comes the heteromorphic haplodiplontic life cycle. So, here we see there is again the mature sporocyte, and then you have the female and the male gametophytes, but they are morphologically different, they are not same. So, that is why they are heteromorphic haplodiplontic life cycle, but in a similar way there will be meiosis from the mature sporocyte, which will produce the gametophytes, male and female, which in turn will give rise to the gametes, and then the fusion of the gametes giving rise to the zygote, and finally the thallus. So, the sporophyte has elaborate development compared to the gametophyte. Examples include laminarales and desmeritales. Then there is a haplobiontic life cycle. Here you can see the haploid phase is dominating and the diploid phase is comparatively smaller. In the case of red algae Nemallion, it exhibits two haploid phases and a diploid zygote, making it a diphasic haplobiontic life cycle or haplontic. On the other hand, the red algae consists of three haploid phases: the main gametophyte, carposporophyte, and Catrencia phase, with the zygote as the only diploid stage. Therefore, this life cycle is referred to as the haplontic. The main gametophyte produces gametes. This is the gametophyte which is producing the gametes, which then fuse to form a zygote that undergoes meiosis and develops into carposporophyte. So, after this fusion, this zygote will be forming these carposporophyte. The carposporophyte will be producing the carpospores, which germinate to develop into the normal gametophyte, as you can see here. The next cycle is a haplodiplobiontic life cycle, also known as the diplohaplontic life cycle. It is seen in certain members of red algae like Polysiphonia, and involves three phases, with two being diploid and one haploid. The main plant body, the gametophyte, produces gametes, and the zygote is as usual formed through the syngamy. So, you can see here the haploid phase and the diploid phase, and you have here certain specialized spores called tetrasporophyte or tetrasperangium, which produces the tetraspores, and then this undergoes formation into gametophytes, which gives rise to gamete, the female gamete, a carpogonium, and the spermidia, the male gamete, which fuses to form the zygote, which is giving rise to the carposporophyte, which produces carpospores, and then finally giving rise to the tetrasporophyte. So, this is the haplodiplobiontic life cycle. Let us now discuss about the life cycle of fungi. We will be discussing about the yeast and the moles one by one. So, let us try to know about budding, which is a key feature of yeast's reproductive strategy. Particularly Saccharomyces family reproduce by budding, a form of asymmetric cell division. You can see here the mother cell is bigger and the daughter cell is smaller. This contrasts to a classical cell division where both resulting cells are typically equal in size, and in the case of bacteria we have seen. In this process, mature mother cells form smaller daughter cells which grows to about 2/3 of the mother size in Saccharomyces cerevisiae. Once the bud reaches this size, cytokinesis occurs separating the cell's cytoplasms. Some yeast species undergo a major morphogenic transition known as dimorphism, which involves switching of budding cell variants to pseudohyphal growth, which is characterized by elongated cells, as you can see here, forming chains under unfavorable conditions. Yeast can transition to a sporogenic form from budding cells, pseudohyphae or hyphae. Spore formation generally follows meiosis, producing meiospores through endobudding, where multiple nuclei within a shared cytoplasm becomes separated, similar to the transition from a multinucleated syncytium to individual cells. In Saccharomycetaceae, meiotic division leads to the formation of tetrads, 4 spores, while in species like Clubomyces, additional mitotic divisions increase the number of nuclei, resulting in asci containing dozens of spores. Each species show great variability in the number, shape and size of their spores depending on the genus and environmental conditions. Another mode of reproduction in yeast is through mating. So, yeast primarily reproduce asexually, but can also produce sexually. Mating involves fusion of two haploid cells of opposite mating types, forming a diploid cell through cytoplasmic nuclear fusion. This process requires mutual signaling and synchronization of cell cycle stages. The mating response triggers cell cycle arrest, enabling the fusion steps that lead to diploid formation. So, here the mating has occurred, and then these enters the diploid vegetative life cycle, and then again due to sporulation these are released and it comes into haploid vegetative life cycle. Now, let us discuss about the sexual reproduction in Zygomycetes, which is a fungi. It occurs by both sexual and asexual process. So, this is the asexual reproductive cycle of Zygomycetes. So, these Zygomycetes primarily reproduce asexually through the formation of sporangia from the sporangium, which is a sac-like structure that produces numerous spores which are haploid. These spores released into the environment and germinate to form new mycelia, the vegetative structure of the fungus. Asexual reproduction allows for rapid colonization as spores can spread widely and grow in favorable conditions. Now, let us look into the sexual reproduction of Zygomycetes, which occurs when compatibility between different mating types are established. So, Zygomycetes have two mating type strains or types, the positive mating type and the negative mating type. Now, what happens when a positive mating type and a negative mating type are nearby? Gametangia from the two mating types will be growing towards each other and fuse in a process called plasmogamy. So, this one is getting fused over here as you can see. The diploid zygote, this results in the formation of an zygote which is actually diploid. These diploid zygote then undergoes meiosis to produce haploid zygospores, which are highly resistant and can survive adverse conditions. When environmental conditions improve, the zygospores will be germinating to form new mycelia and thereby completing the life cycle. So, these two mating types approaching each other, fusing, forming the zygote, which undergoes later on a diploid cell and undergo meiosis, releases the haploid spores, which will germinate under favorable conditions, thereby completing the life cycle sexual reproduction in zygomycetes. Overall, we can see that there is a sexual reproduction cycle over here in zygomycetes and a sexual reproduction cycle over here. It can also enter from the sexual reproductive cycle to the asexual reproductive cycle through this common linkage, the formation of the spores. So, this is the overall typical life cycle of zygomycetes or zygomycota. Now, this is the typical life cycle of Ascomycota, which also reproduces by sexual and asexual reproductive methods, and certain things here are also similar. Now, let us examine in details how the reproduction in Ascomycota happen, as in the case of Zygomycota. So, sexual reproduction in Ascomycetes begins with plasmogamy, as you can see over here, where haploid hyphae of opposite mating types, positive and negative, as in the earlier case, will fuse and forming dikaryotic hyphae. So, this is the dikaryotic hyphae which is getting produced as a result of this plasmogamy. Here. So, the karyogamy will follow, where the nuclei fuse to create a diploid twisted nucleus. Here this karyogamy is happening, and then meiosis will occur, reducing the diploid nucleus to 4 genetically unique haploid cells. So, here these are the meiosis products, these are the 4 unique haploid nuclei happening over here. Often an additional mitotic division occurs, resulting in 8 haploid nuclei. So, from these 4 it will become 8, and that is the ascospore or the ascus. These nuclei are enclosed in a sac-like structure called asci, which develops from specialized ascogenous hyphae. The asci eventually releases haploid ascospores for the dispersal. So, these are all getting released for dispersal, and then they germinate and then give rise to the 2 mating types which fuse by plasmogamy and completes the life cycle. Within the ascus, each haploid nucleus typically undergo 1 mitotic division, resulting in 8 ascospores. Though the number of ascospores can vary between species, upon maturity the ascospores are discharged through mechanisms such as rupture of the ascus wall or expulsion through an opening at the ascus tip. Once released, the ascospores disperse to new environments where they can germinate and develop into a new hyphae. So, with this we come to end of this lecture. Thank you for your attention. We will be continuing this with some other organisms in the next lecture.